Now we come on to the post-absorptive state, which is generally considered to be the time beyond the fed state, so anything after those 4 hours or so after eating a meal. And this is when you've digested your meal, it's been absorbed and the nutrients metabolized, so there's no more abundance of macronutrients from that meal circulating around the body. So you've essentially run out of your supply of fresh fuel and have to rely on the stores put there earlier. The first thing you need to remember is that the body needs to continue producing energy all the time, and glucose is always the preference because the brain, under normal conditions, only functions on glucose, not fatty acids or amino acids. So blood glucose is required to remain constant to provide the brain with energy. And when you're in the fasting state, where can your body get glucose from? So we get it from the liver's glycogen stores, and this is glycogenolysis. That's the process which is the lysing or splitting of stored glycogen to release glucose for use in the body. But there's also a finite amount of glycogen stored in the liver, maybe 12 to 18 hours worth. So if you're fasting and not eating for a long time, the body won't want these glycogen stores to run down too much, as they're so important. So preservation of glycogen and stored glucose and so on is a key aspect of how your metabolism is functioning in this post-absorptive state. If glycogen gets low, then the body will upregulate its mobilization and use of fatty acids as a source of fuel, and the blood concentration of fatty acids will rise, and as that happens, the cells and tissues will take up more fatty acids and use them as energy. And remember that we can also synthesize new glucose from amino acids, so muscle tissue or free amino acids floating around in the blood can be deaminated, as we've mentioned before, and the carbon skeletons of glucogenic amino acids can be used to synthesize glucose in the liver via the process of gluconeogenesis, which is the synthesis of new glucose from those glucogenic amino acids. Remember, they're the two main types of amino acids, glucogenic and ketogenic amino acids, and it's the glucogenic, as I'm sure you can tell by the name, are the ones which can go on to synthesize new glucose. So I hope you're understanding the common sense logic behind all this. Let's have a closer look at glucagon, which is essentially the opposite hormonal partner to insulin. So it has sort of the opposite effects. One of the key things when the body finishes absorbing carbohydrates is it must maintain glucose levels, particularly for the brain, and also, of course, red blood cells, which have no mitochondria, so can only undergo anaerobic glycolysis, so can't use fats for energy, for example. What happens is we mobilize our stores of glycogen to synthesize new glucose, which is the storage form of glucose, and it's the hormone glucagon which stimulates this process. As insulin drops as a result of falling blood glucose levels, because remember we're now in the post-absorptive state, the fasting state, our glucagon levels start to rise. So remember that insulin is produced by the pancreatic beta cells, while glucagon is produced by the pancreas's alpha cells, so that's easy to remember, I think. And that's secreted about 4 hours after a meal as plasma glucose drops. Also, if blood sugar levels drop, then adrenaline is stimulated, which also results in a rise in glucagon. And this is believed to be a survival mechanism, so when we get hungry, and as we've evolved, in these hunter-gatherer lifestyles, and we get hungry and we're in need of food, then we get that perk up from the adrenaline to go out and hunt or gather up food. And from a practical modern day point of view, consider that if you go to bed hungry, or you send the children to bed without dinner because they've been naughty, and your body's upregulating its release of adrenaline, how well do you think you will be sleeping? How deeply do you think you'll be sleeping? What will be the quality of your sleep, or will you be able to drop off at all? So consider that in the evening you do need to eat something to prevent that adrenaline rise from being hungry. Unsurprisingly, glucagon is inhibited by insulin, and it acts on the liver only to regulate metabolism. So the metabolic effects of glucagon in the liver. So glucagon has its effects on the liver to break down glycogen to stimulate the synthesis of glucose. We touched that already. It stimulates the synthesis of glucose from glucogenic amino acids, which will be deaminated, and the carbohydrate skeleton, which will be used to synthesize glucose. So that's gluconeogenesis, you know that. And it also stimulates the uptake of amino acids in the liver for gluconeogenesis. So you can see that where insulin stimulates the uptake of amino acids in muscle to build more muscle tissue, glucagon will stimulate the uptake of amino acids in the liver so that it can synthesize glucose. So perhaps unsurprisingly, glucose also inhibits glycolysis in the liver, as well as stimulating the oxidation of fatty acids and synthesis of ketone bodies. With regard to ketone bodies, they are one of the molecules which saves our glucose stores. As I mentioned before, glucose is quite precious in the body, and the body tries to conserve its stores of glycogen. So it will be conserved if we go too long without food. And if we use both fatty acids and amino acids to save glucose stores, this is one mechanism, the main mechanism that can go around to preserve or prevent the overutilization of glucose and glycogen and breaking down glycogen stores. And we get more energy from fat, as you know, and that saves our glucose stores. We synthesize new glucose from amino acids, and that saves our glucose stores. But then the body starts to get worried about breaking down a lot of proteins, so to synthesize glucose, because of the toxic byproducts, remember, involved in gluconeogenesis, so it uses ketone bodies and makes more use of fat stores. So the liver increases its production of ketone bodies, which circulates in the blood and can be used as an energy source in cells. The longer you go without food, the more the body will switch to using ketones for energy. To the extent where in conditions such as diabetes, where you can't get sugar into the tissues, or in starvation when you get ketoacidosis, which is where the level of the acidic ketone bodies in the blood gets so high it affects the pH of the blood and it overwhelms the body's buffering mechanisms. And so you're not getting an acidosis situation, which will happen in uncontrolled diabetes, for example. And if the situation with insufficient glucose becomes desperate, even the brain will start to convert energy use to ketone bodies in order to spare glucose. But here we're looking at 2 weeks or more without food, so it's not a normal situation which we'll come across. Let's just take a closer look at gluconeogenesis, mainly from amino acids, but there are other substrates as well. Gluconeogenesis is essentially the reversal of the glycolytic pathway. So glycolysis, the reversal of glycolysis, except for the final step, because that final step of glycolysis is irreversible. So instead, oxaloacetate, which is the starting point of the Krebs cycle, you can see in the diagram, is diverted and it's decarboxylated and enters the glycolysis pathway just up from pyruvate, and then the glycolytic pathway is reversed. So let's have a bit of a closer look at that. Synthesis of glucose, reversal of the glycolytic pathway, final step of glycolysis is irreversible, so bypassed via oxaloacetate. And I said, remember, it's mainly from amino acids, but there are other intermediates from which gluconeogenesis can occur from. Okay, so this is the final step of glycolysis, being the conversion of PEP, on the diagram you see there, to pyruvate, pyruvic acid. As I said on the last slide, it's irreversible. So if you want to get pyruvate and reverse glycolysis to synthesize glucose, you need to convert pyruvate to oxaloacetate, if you follow the light blue line down there. And then oxaloacetate is then decarboxylated, and you can see from the diagram that it can then be converted back to PEP. And that's decarboxylation. It requires vitamin B1, so thiamine, and the carboxylation of pyruvate to oxaloacetate requires the B vitamin biotin, as well as manganese. You can see in the little yellow diagram there. So again, you can see the importance of the B vitamins in the energy production process. And all of these intermediates, and all of the intermediates from the Krebs cycle, can be used for gluconeogenesis, like on the previous slide, because all they have to do is take one quick spin around the Krebs cycle, and as you know, because it's a cycle, they will end up as oxaloacetate. So where's that diagram before? There we go. You see oxaloacetate being the start of the Krebs cycle. And then they can then be converted to PEP and then to glucose. And that's why the glucogenic amino acids are those that can be converted to Krebs cycle intermediates, because they go around the Krebs cycle to be metabolized to oxaloacetate and then into the gluconeogenesis pathway. Right, absorptive tissue in post-absorptive state. So let's just explain what this is all about. Because you have a drop in insulin, you get increased activity of the lipase enzyme, which catabolizes triglycerides to free fatty acids floating around, and glycerol, of course, and then releases them into the circulation. So with low insulin, you get an increase of enzyme activity, and thus more fatty acids mobilize from the adipose tissue. And we know from our previous slides that in the starvation state, the fatty acids will be converted to acetyl coenzyme A, which will then be siphoned off away from the Krebs cycle to synthesize ketone bodies. Again, if blood sugar drops and you have the resultant adrenaline stimulation, that will also trigger the synthesis of lipase to mobilize fatty acids from triglycerides, and as the fatty acid part of the triglycerides has been used, the remaining glycerol part will enter the gluconeogenesis pathway in the liver. So coming back to this chart, as we go longer and longer without food, the blood glucose starts to fall, as you can see, as it sort of approaches 24 hours and so on. And consequently insulin falls, and instead we get insulin's opposite partner, glucagon, in orange, start to rise. As we mobilize our glycogen stores in the liver, so the liver glycogen levels descend as it's converted into glucose. The blood concentration of fatty acids starts to increase as they're mobilized from adipose tissues, from glucagon, and also the ketone body starts to rise, although it starts a little later than fatty acids increase, because as you can see, ketone levels will increase those of fatty acids after a few days of not eating, because the fatty acids are still being used as a source of energy. And as we get into the starvation state more, the ketones will continue rising and rising. So let's have a look at the summary of the alternative fuels used to spare glucose. So the hepatocytes, the liver cells, oxidize fatty acids and amino acids to generate ATP. The heart is not fussy, so it will oxidize fatty acids, ketones and also lactate. The muscles and kidneys can also use fatty acids and ketones, and as you know, the brain is the primary user of blood glucose, but in extreme fastings or starvation states it can also use ketone bodies. So from this you can see that the key organs in the body are quite capable of using alternative sources of fuel. And this diagram simply might be worth you studying just to get a visual idea of some of those processes we've discussed, and might help you if you're a visual learner. And finally, I know that sometimes when you hear about the various metabolic reactions taking place, it helps to have an understanding of where they are taking place, in which part of the cell and in which tissues, so that you can picture it in your mind. So this table gives you an overview of the location of the reactions. So I hope you've understood what we were discussing in the metabolism lecture. I do recommend, if you're not that clear on it, you go back through it and re-listen to it, and read your notes, and certainly read around in your textbooks, and I'm sure you'll get a good grasp of it, because it's one of those topics which will just keep coming back again and again. And to understand metabolism, not to be afraid of it, understand metabolic pathways and how they fit together and how they relate to each other and everything, is going to be real gold dust for you later on as a practitioner, and later on in your studies on the nutritional therapy course.